6. So we're going to talk about the different types of RNA and what those different types do, give you a roadmap of where we are. In the chapter 1, we were talking about molecules and mostly how they carry information. Now we're going to talk about cells and how information is transmitted in cells. So we're in chapter 2. We'll see cells again in chapters 5, 8, 11, and 14. All right. Cells are the basic unit of life, and cells are made up of macromolecules, those big molecules we talked about before, lipids, carbohydrates, proteins, and nucleic acid. There are what is required to have a cell is you have to have some kind of DNA sometime in its life cycle. Sometimes mature cells kick out their DNA. Also RNA, and then protein, lipid, and carbohydrate. There's an aspect of modern biology called synthetic biology that is trying to figure out exactly how many genes do you need in order to be a cell, in order to be an organism, and can we make our own custom cells from scratch? So do we understand enough about these rules that we can make them from scratch? And that's called synthetic biology. To give you an idea of how it works, if you look at an egg, this is a hard-boiled egg. There's some macromolecules in here. Yolk is primarily lipid. Albumin, the white of an egg, is a type of protein. This is why eggs are full of protein. And when this was not boiled but alive, so not this whitish and yellowish cakey color, but instead a live egg, there would be a little tiny cell sitting on top of that yolk, so small you couldn't see it, and that would turn into a chicken. And all the instructions on how to make that chicken are in the DNA of that little tiny cell. And this stuff here, the albumin and the yolk, will act as fuel to get the chicken started on the first part of its egg until it hatches. And that's an example of central dogma. Dogma is a word that is used to describe something that we think is true, something that everybody agrees on as true. And the central dogma of biology is that DNA codes for RNA, RNA codes for protein. So the DNA in that little tiny egg cell that's going to become a chicken is got all the information to code for the chicken. That information has to be decoded from the DNA and turned into RNA, and then that RNA has to be turned into protein in order to make the chicken. So you can think of DNA as an instruction manual or sort of like a cookbook. RNA is temporary scratch paper that you write down the part of the cookbook that you're interested in. Let's say you're going to make brownies. You write down the brownie recipe. There are a lot of other recipes in that cookbook, but you're just interested in making brownies, and maybe you write it on a whiteboard or a little post-it note that's a temporary thing that you write down the ingredients for those brownies. And then proteins are actually the brownies. That's the machine that is made from the instructions in DNA, translated through the RNA, and then turned into protein. DNA to RNA is actually called transcription, and RNA to protein is called translation. So you go from a single cell to a chick, and all the information is in on how to do this from a single cell to a chick is recorded in that DNA. But when you're looking at the chick, you're not looking at DNA. Most of what you're looking at is protein. And to illustrate that, here's Greta Thunberg from Time Magazine, Person of the Year. When you were looking at another person, their skin is protein, their eyeballs are protein, their hair is protein. Most of what you're looking at when you're looking at a person is protein. So it's not DNA, it's not RNA. It's the what those things turn into. So DNA is first converted into RNA by an enzyme called RNA polymerase, and that is called transcription. RNA is then converted into protein by another type of machine called a ribosome inside your cells. We'll talk about those today. And those are made up of RNA and protein, and that process is called translation. And every cell in Greta, every cell in you has the same DNA, but not all of that DNA is transcribed into RNA at the same time, just like you wouldn't necessarily make every recipe in the cookbook. You can choose which recipe you make. So you have genes in your DNA that code for a nose. You have genes in your DNA that code for eyelashes. And the cells that give rise to eyelashes translate those, transcribe those genes to make eyelashes, and then translate that RNA to turn into eyelash protein, keratin, on your eyelashes. And those eyelashes aren't growing on your nose because different genes are transcribed on the cells that make up your nose. And those different genes are transcribed and then translated into different proteins, so you get something that looks like a nose, not like eyelashes. So every single cell in your body has the same DNA, but it's not read the same. So you have the same recipe book in every cell, but you're opening up and reading a different recipe and actually making a different recipe in all those cells. And once again, DNA converted into RNA by RNA polymerase, that's called transcription. RNA converted into protein, that's called translation. These two words sound similar. You may see me mixing them up every once in a while, and I'll catch myself on that. It's important that you know the difference. So RNA to protein is translation. DNA to RNA is transcription. So again, if we go back to our recipe analogy, when you transcribe what's in the recipe book onto a Post-it note, that's transcription, that's making RNA. And then when you actually translate what you wrote down into a cupcake or a brownie, that's translation. Okay? All right. How do scientists study RNA, and what sorts of RNA are there? There's a technique called gel electrophoresis. This is a picture of a gel. And in gel electrophoresis of nucleic acids, DNA and RNA, they use a gel that is made up of carbohydrate, agarose, agar, you might be thinking of from seaweed. And that you can melt and it will turn into a solid jello-like consistency stuff that is full of holes, pores. And then when you apply a sample of DNA or RNA to an indentation in your gel, and then you can pull that sample through the gel using electrophoresis, electricity. You put the positive pole down here, the negative pole up here, and all those phosphate groups on the DNA and RNA make DNA and RNA negative, and so it pulls it through the gel. As if you have a big piece of nucleic acid, a big piece of DNA or RNA, it's going to get stuck in the pores sooner. So larger things get stuck up here, smaller things get separated down here. So this separates things according to size, and you can get an idea of different sizes of your sample as it travels through the gel. You can also label the nucleic acid with a fluorescent dye that shows up bright when there's DNA. So there you see bright bands here. This is actually DNA or RNA. This is actually RNA. So this MW stands for molecular weight markers, and then these are 2 samples of RNA. So the molecular weight markers, this is actually a lame picture from your book. They should be much brighter than this, but you can see individual bands here. These are pieces of DNA, typically DNA, because DNA is more stable and it's just cheaper to use, that have known sizes. So if I know that this, for example, this band right here is 500 base pairs long, then if I have another sample that I don't know what size things are right next to it and it migrates to the same place, I know that that thing is also 500 base pairs long. So that's what those markers are for, is just to tell you how big things are in your unknown samples. So in your unknown samples, you've got these bright bands showing up, and you also see some smeary sort of bands here, very faint pattern, and then another smear down on the end. So this would be bigger stuff, middle-sized stuff, small stuff. All right, what is that stuff? Well, the most abundant RNA in all of your cells is RNA that goes into making the ribosome. And the ribosome is the translation machine. It's the machine that takes RNA and reads that RNA and translates the information and turns it into protein. That ribosomes are made up of a large subunit and a small subunit. This is the cartoon version of it. This is what it really looks like. And the ribosome large subunit is made up of both ribosomal RNA, shown here in red, and also ribosomal proteins. So the ribosome is made up of protein and RNA. Small subunit, same thing. It has ribosomal RNA in it and also proteins. And together they come together to form a ribosome. The small and large subunits come together when they are going to make a protein, and when they are done making a protein, they come apart. You have just gobs and gobs of ribosomes. They are the most common thing that you would see inside a cell. It's pretty big as cell structures go, big enough to see with an electron microscope. Not as big as something like a mitochondrion, which we'll talk about later, but big enough that you can see it as a little typically black dot. And how many does a cell have? Like 10 million per cell. So cells make lots of protein. They make protein all the time, and these little protein making machines are all over the place. And the protein making machine, the ribosome, is made up of itself. The machine is made up of both ribosomal RNA and ribosomal protein. Okay, so what can we learn and how do we know about what kind of RNA makes what and how is it involved in the translation process? So this is an experiment where they separated RNA according to size. So they put it through a centrifuge, similar to the experiment we talked about in the last podcast. And big stuff shows up on this part of the graph, small stuff shows up on this part of the graph, and they can look at different kinds of RNAs. So ribosomal RNA is here, and you see 2 peaks for the large subunit and the small subunit, and then transfer RNA, a different kind of RNA, shows up later. It's smaller. So 2 sizes of ribosomal RNA, large subunit and small subunit sizes of ribosomal RNA, and then transfer RNA is pretty small. In trying to find out what happens and trying to prove that what is the RNA that carries amino acids, they added radioactive leucine. Leucine is an amino acid, and that radioactive leucine they can plot separately as this purple line, and it shows up in the same peak as the transfer RNA. So based on that, we know that the leucine is associated with transfer RNA, and now we know, based on experiments like this, that transfer RNA is the RNA that carries amino acids. And they bring the amino acids to the ribosome. Okay, so transfer RNAs look like this in real life. Each one of these are individual atoms. The gray atoms are carbons. The red atoms are oxygens, and the blue atoms are nitrogens, and orange is phosphorus. These are often drawn just because it's easier to draw like this as a flattened shape, and you can see it sort of has a T shaped in there. That's not how they got their name. The T stands for transfer RNA. It transfers amino acids to the ribosome, but they also sort of look like a T. The tRNA is a single strand of RNA that is folded up on itself, and this is base pairing. So this is a G with a C or an A with a U. So you might remember in a previous podcast I was talking about the bases in DNA, A, T, C, G, and in RNA it's A, C, G, and instead of T, it's U. U for uracil. So in RNA, A binds with U instead of A binding with T, like it does in DNA, and then G still binds with C. So this is a G with a C or an A with a U stuck together like that, and it gives it this shape, which in 3D comes out to that shape. So on the 3' end, there's a hydroxyl end. Remember we talked about the 5' and 3' carbons on that sugar in the middle of nucleic acids. On the 3' end, there's an OH group, and that attaches to amino acids. Okay? And then the other important part of a transfer RNA is an anticodon. It's a group of 3 bases, 3 nucleotides that are going to bind to the messenger RNA, which we'll talk about next. On the messenger RNA, it's called a codon. One group of 3 is called a codon. And on the tRNA, the group of 3 that binds to the codon is called the anticodon. Okay, so the amino acid attaches up here, and the anticodon attaches the mRNA down there. Okay. So can we see other things on this gel? So we know that these are the ribosomal RNA bands, the ribosomal RNA that make up the RNA portion of a ribosome. And by far, because there's so many ribosomes in a cell, if you extract RNA out of a cell, you're going to get lots and lots of ribosomal RNA. Okay? The RNA in this blob down in the bottom, that's transfer RNA. It's small. And this faint smear that you see in between is messenger RNA. And messenger RNA is all kinds of sizes. Could be big, could be small, and it depends on the gene that it came from. So when DNA is converted to RNA, that's called transcription. And if the gene that is being read and turned into RNA is a big gene, then it's going to be a big long molecule, big long messenger RNA. If it's a small gene, it's a small molecule and a small messenger RNA. So messenger RNA comes in all kinds of different sizes. Ribosomal RNA is always typically the same size and is pretty stable. And transfer RNA is always small. Okay. This is a different graph than the one I showed you previously, so not the same as this graph. Okay, it's a different experiment. Okay, but the same idea that they're trying to figure out where things are in the cell. Okay, so again, you have 2 peaks of ribosomal RNA associated with small and large subunits. What in this case, it's a this is RNA, ribosomal RNA not associated with a ribosome, and when it is associated with a ribosome, it gets it shows up as bigger on this graph, small to big in this case. The other one was the opposite, so small here, big here. Okay. This time they're adding a radioactive uracil. Uracil is a base that is specific to RNA. So they wanted to find out for newly made RNAs, and not DNA, can't be DNA because DNA doesn't have uracil, where do they show up? Okay, so you have small RNAs and then large RNAs here. The RNA is measured by UV absorbance, and they can see, okay, you have a peak of ribosomal RNA that is relatively smaller, and then another peak that is associated with a ribosome. And then when we looked at the radioactive RNA, after this would be RNA that is newly made. So the cells are chugging along and doing transcription, then all of a sudden you give them some radioactive uracil, and only RNA that is made after you gave them the radioactive uracil will show up on this graph. If it's RNA that has already existed, it won't show up as radioactive because it's not newly made. So the ribosomal RNA is not showing as radioactive because it's stable, it's sitting in the cell, it's already part of a ribosome, and so it's not going to be radioactively labeled. But what does get radioactively labeled is new transfer RNAs. They get made up, and they are small. And then also messenger RNAs. So as the cell reads the DNA, converts it into messenger RNA, that messenger RNA eventually is going to go to the ribosome and be read by the ribosome. And you can see that this mRNA gets labeled. The mRNA can be all different kinds of sizes depending on how big the gene is that is being read. And that mRNA is associated with the ribosome. So the transfer RNA brings amino acids to the ribosome, and the messenger RNA also gets moved to the ribosome to be read by the ribosome. The messenger RNA carries the message of the DNA and says, okay, here is the protein I want you to make based on the DNA code. And it's going to put amino acids together in a certain order according to what is dictated by the mRNA. And the ribosome is the machine that is going to put them together. And then the amino acids get carried there by the tRNA. Okay, so again, the ribosomal RNA makes up the ribosome, which is the machine that's going to do the translating. The mRNA is a result of transcription of DNA. So the gene gets read by RNA polymerase. RNA polymerase takes the information in DNA, converts it into messenger RNA. That messenger RNA then travels to a ribosome, where the ribosome is going to read the information on that transfer RNA, and the information is going to tell the ribosome what amino acids to put together to make a protein and what order should it put those amino acids together in. And the amino acids get carried to the ribosome by a tRNA. Okay,